Welcome, everyone. I'm excited to kick off our presentation today on sampling the air microbes. This is the first slide, so I want to warmly welcome you all and introduce our main topic. We'll be exploring how we collect and analyze microbes present in the air around us. Over the course of this presentation, we'll look at the methods used, why this research is important, and what we can learn from it. So let's get started. When we talk about impinge, we're referring to the effect something has on another, often causing problems by limiting it in some way. In the context of air sampling, impingement methods are used to isolate microorganisms from the air. There are two main types: solid impingement and liquid impingement. Both methods are designed to help us enumerate, or count, the microorganisms present in the air, but it's important to understand their limitations. They don't collect and count every single microorganism in the sample. Some cells get destroyed during the process, and others simply pass through without being captured. So, while these methods are useful, they do have their drawbacks, and the key is understanding that they provide an estimate, not an exact count of all airborne microbes. This connects to our overall focus on accurately assessing microbial presence in air environments. Let's talk about impingement in liquids. In this method, air is drawn through a very small opening or tube, which creates a fine bubble stream that bubbles through the liquid. As the bubbles move through the liquid, they trap the microorganisms present. This is a useful way to collect microorganisms from the liquid medium. After capturing these organisms, we take aliquots of the liquid and plate them, meaning we spread small amounts onto culture media to determine the microbial content. This technique is important because it allows us to efficiently sample and analyze microorganisms in liquids, which ties into the broader theme of detecting and quantifying microbes in different environments. Today, I'll be discussing the impingement on solids method. In this technique, microorganisms are collected directly onto the solid surface of an agar medium. Essentially, the microorganisms in the environment come into contact with the agar surface, and as they do, they adhere to it. Over time, as the microbes settle and multiply, colonies start to develop right on the medium where they initially impacted. This method is quite useful because it allows us to observe the growth of colonies directly on a solid surface, making it easier to identify and count them. It's a straightforward way to assess microbial presence and activity on solid surfaces, which is a key part of understanding microbial contamination and behavior. So, in summary, this technique captures microorganisms on agar where they land, leading to colony formation that we can then analyze further. All right, let's talk about active sampling methods. These are techniques where we take a proactive approach to selecting samples, often aiming to gather the most informative data possible. The idea is to strategically choose samples that will give us the greatest insight or improve our model efficiently. Now, contrasting that with passive sampling methods, which are more about collecting data without specific targeting. With passive sampling, we simply gather data as it naturally occurs or is easily accessible, without trying to influence which samples we get. Both approaches have their place, but in the context of our overall goal, optimizing data collection and model performance, understanding the difference helps us decide when to be proactive and when to go with the flow. Active sampling can be more resource-intensive, but often yields better results faster, while passive sampling is simpler but might require larger datasets for the same level of accuracy. Let's take a closer look at air samplers, starting with sedimentation and slit samplers. Sedimentation methods involve collecting aerosol particles on a sticky surface, like a Petri dish containing agar or glycerol. This approach allows particles to settle naturally over time, making it a simple and effective way to sample aerosols. On the other hand, slit samplers work differently. They direct sampled air through a narrow slit onto a rotating collection surface, often an agar medium Petri plate. The rotation is intermittent, which means each impacted area on the surface corresponds to a specific volume of air sampled and a particular time segment. This setup allows us to collect a time series of samples, giving us valuable information about how aerosol concentrations change over time. The agar medium in the Petri plate is crucial for cultivating bacteria, or other microorganisms present in the air. So, to sum up, sedimentation is straightforward and relies on natural settling, while slit samplers provide a more controlled, time-resolved sampling method. Both techniques are vital tools in understanding airborne particles and microorganisms, tying into our overall goal of monitoring and analyzing air quality and microbial presence. Now, let's take a closer look at the air samplers, specifically the stacked sieve or Anderson-type sampler. This device consists of 6 stages, each being a perforated plate positioned above a Petri dish. The interesting part is that the diameter of the air passageways gets smaller at each subsequent level. This means that as air passes through, it progressively captures smaller particles. Essentially, this setup allows us to classify and collect particles based on their size. It's particularly useful for sampling bacteria, which are often in the range that this device can effectively trap. Additionally, it can be adapted for virus sampling by collecting viruses onto a sticky surface within the Petri dish. Overall, this sampler is a valuable tool in our efforts to monitor and analyze airborne microbial contamination. Let's talk about air sampling using filtration methods. The basic idea is to pass air through a membrane filter that has a small enough pore size to trap aerosol particles. This way, we can effectively collect the particles suspended in the air. After the collection, there are a couple of options. We can either plate the filter directly to culture any microorganisms or wash off the particles for further analysis. However, there are some challenges to keep in mind. Desiccation or drying out of the collected particles can affect their viability or appearance. Additionally, if the washing process isn't efficient, we might not recover all the particles, which could impact our results. Now, an important tool in this process is the open-face air filter cassette with a cap. It's a straightforward, practical device that allows us to securely hold the filter during sampling, ensuring we get a good sample while minimizing contamination. Overall, filtration methods are a vital part of air sampling because they enable us to capture and analyze airborne particles effectively, which is crucial in assessing air quality and safety. Now, let's take a closer look at air samplers that use electrostatic precipitation. Essentially, these devices draw air over collection plates that are electrically charged. The key here is that charged particles in the air are attracted to these plates, which are either positively or negatively charged. The surface of these plates is wetted, which helps in capturing the particles effectively. Once the particles stick to the plates, they're washed off into a circulating collection fluid. This process makes it easier to analyze the particles later on. What's important to remember is that electrostatic precipitation is a highly efficient way to collect airborne particles, especially when dealing with very small or elusive particles. This method ties into our overall theme of understanding air quality and how we can monitor it effectively. It's a clever use of electrical forces to improve sampling accuracy and efficiency. Now, let's talk about liquid impingers, which are a type of air sampler used to collect particles from the sampled air into a liquid medium. One common example is the all-glass impinger, or AGI. The way it works is quite interesting. Particles are drawn through a small orifice that significantly increases their velocity. This acceleration causes the particles to impinge on the bottom surface of the container, effectively scrubbing them into the collection fluid. This method allows us to trap airborne particles efficiently, but it's important to remember that there are some challenges. Excessive cooling and evaporation can occur during sampling, which can affect the integrity of the collection. Additionally, leakage can be an issue, meaning particles might not be retained properly if the device isn't sealed well. Overall, liquid impingers are a useful tool for collecting airborne particles, especially when combined with proper handling to minimize those issues. They fit into our broader theme of understanding and improving air sampling techniques to ensure accurate measurements. Now, let's talk about air samplers, specifically the high-volume liquid cyclone scrubber. This device works by drawing in air at high speeds, and as the air passes through a gradually narrowing helical pathway, the particles in the air are forced to collide with the container walls. This impaction helps to effectively remove particles from the airstream. The collected particles are then captured into a recirculating collection fluid, which is maintained by a pump. This method is quite efficient for sampling large volumes of air and capturing airborne particles, making it a valuable tool in environmental monitoring and workplace safety. Overall, the cyclone scrubber exemplifies how we can use physics and engineering principles to improve our ability to measure and control airborne contaminants.